So today we're going to cover what's new on the UniProt website, features and improvements for 2026. Um, for a little about this webinar guide. So this from this webinar, um, it's geared for all UniProt users. If you have undergrad biology, that would really help in terms of understanding the maybe the terminology and the concepts of the website, but it's not really required. Today we'll cover new data annotations that we included into the UniProt website, and then the improvements in UniParc and the Proteomes namespaces that we have. We'll also cover the ID mapping, BLAST, and some tools that we have um, existing on the UniProt website that most of us will probably be using. And um, after each section, we'll have a little demo uh, to show the changes and how to use it on the website. So please stay for those. And then if you have any questions, you are welcome to put it up on the Q&A part. So what you'll be able to do after this session, so first is to obviously navigate recent additions that we had, including the sequence annotation pages, AlphaFold structure views, and variant viewer improvements. You will be able to find new annotations. So these include the PTMs in the feature viewer and expanded help documentation. And you will also be able to use the improved search and ID mapping across UniProtKB, UniParc, and Proteomes. Here's the agenda. So we've divided this presentation into 4 parts. So the first will be dealing with the changes in UniProtKB. I will be introducing you to these. For part 2 and 3, which we will cover UniParc and the tool improvements, Swaathi will take over in explaining the improvements and the reasonings behind it. And then at the end, we have a Q&A where Antonia will also help us as a curator to answer any questions related to biology, while Swaathi and I can answer some technical questions and how to navigate the website and things like that. So you're welcome to once again post any questions that you have while we listen to this webinar. Okay. Before we start, a quick refresher on UniProt. So we have UniProtKB, UniParc, and Proteomes under the UniProt website. UniProtKB is the one that our users spend the most time on it. It's consisting of the reviewed SwissProt entries and unreviewed Tremble entries. These are primarily reference proteomes and other selected entries that exist in UniProtKB. The others are stored in UniParc along with the entries that we have in UniProtKB. So UniParc is a non-redundant archive of every protein sequence UniProt has ever seen. So it's a historical archive that will contain all the sequences that UniProt ever had. And for the proteomes, it's sets of proteins thought to be expressed by an organism. So here we'll have, you can search for groups of proteins within a taxonomy or within a proteome. And all 3 namespaces here are interconnected. And this is where most of our today's improvements and today's talk will be focused on. A quick overview of what's new. So in UniProtKB, I won't go into too much detail here, but UniProtKB, we've included the points that you see there. In UniParc, we have some taxon and proteome suggestions, updated FASTA downloads, the additions to the entry page, cross-reference handling, sequence annotations. In tools, we have blasts and ID mapping improvements. So yeah, this is just a brief overview. We'll go into detail starting now. So part 1, UniProtKB. The first section that we would like to introduce is the new homolog section inside the UniProt entry page. So this is from the Alliance of Genome Resources Orthology, so AGR. These are curated orthology data for selected organisms from the AGR consortium, and this is now available in the UniProtKB entry pages. It's at the very end of the page, so the last section within the entry page will contain this information. You can see side by side with existing orthology resources, which is easier for cross-species comparison. And it's useful when you're trying to move between the model organism resources and UniProt. So here within, as you can see in the screenshot, there will be, you can see it's a APP orthology. So you have the Musmunculus and Rattus, two different species of Rattus, and then Xenopus, etc. So this is all the things that you could look into the same protein within different species. The next one is the AlphaFold structure views. So previously, we only had the AlphaFold structure viewer, but without the isoforms. So it was only the canonical sequence that was displayed in the structure viewer. Now we have included the isoforms as you can see in the video that's playing. The -1, -2, those are the different isoforms, and the canonical one is labeled as canonical. So you can switch between the isoforms in the structure viewer, and each isoform links to its corresponding isoform in the sequence and isoform section. So you can actually go to the particular sequence or isoform sequence if you want the actual sequence to copy, to use it for BLAST, etc. And another improvement in the AlphaFold complexes is the AlphaFold complexes in the structure viewer. So these show the predicted complex structures surfaced from AlphaFold homodimers, and you can discover the binding partners directly from the entry page. So each complex links back to the participating UniProt entry, which is something that we're trying to improve in the future. But this is a functionality that will be added quite soon. So we decided to just mention it here. And because of the complexes, it'll be a bit ideal for protein and protein interactions questions that you have while exploring a particular sequence. Next would be the card cross-reference showing the resistance mechanism. So we have uh, for bacterial proteomes, we have a card cross-reference that is existing, and the card stands for Comprehensive Antibiotic Resistant Database. And now, before we only had the cross-reference, now we've actually added the resistance mechanism and the link to the actual type of resistance mechanism that a particular protein might have observed. So as you can see in the video, you can find it right there in the protein family group databases with a card, and it also shows the resistance mechanism at the bottom. The next one would be the variant viewer. So this isn't anything new relating to data, but it's a new Canvas implementation. So previously we had the variant viewer under some limitations because it was a different implementation. It was quite heavy. So once a variant viewer or a sequence had more than a couple thousand annotations, it got really sluggish and it was very difficult to navigate. Now we've implemented this via a Canvas method, so it's a lot faster in terms of the interactions and um, the loading in general. So everything that you can do in terms of like zooming in or zooming out, that would be a bit faster and snappier. And hopefully that is a good improvement for all the users and that uses the variant viewer. And relating to that is the feature viewer and the new annotations to the PTM regarding PTM. So we have additional post-translational modification types, including ubiquitination and acetylation. So these are found under the feature viewer, and if you open the PTM tag or track, as you can see in the video, there'll be different types of annotations that has ubiquitination and other acetylation tracks as well. So, and a good point is if you expand the track to multiple rows, you can actually see it won't be overlapped. So all the annotations are clickable, even if they're on the same location. So that is a good way to navigate the annotations. Um, Next one is the improved entry history page. So this includes demerged entries. So in UniProt KB, if you look at the history tab on the very right, there's um, a little explanation of showing all the sequence history and the entry history of that particular accession. And now we have added some more data or more information to show that um, there's a clear view of what changed and when it changed across all the entry versions. We also include demerge entries. So this demerge entries means when one entry is split into multiple different ones. So we now have a dedicated navigable history regarding this one. So for P03401, as you see in this example, it has been demerged into four different ones in UniProt KB, and we all link to it. So you can actually find the ones that you need. And yeah, the inactive entry history is loaded inline, so there are no dead ends, and everything that you want to find from the history tab will be there as well. So here I'm gonna go to a quick demo regarding um, the UniProt KB entries that we've done. So I'll copy that, and then from uniprot.org, I'm gonna search for UniProt KB P04637. So here we have about, again, at the very end of the section, we have the homolog section with the orthologs and paralogs. From here, you can filter by species if you only want to see one of them. You can also go to paralogs to see the gene symbol of TP63, TP73, and see which methods and accounts are there. If we go from here to the variant viewer, this will open up a new tab and you'll have all the variants here, which is 3000, about 3500. So if I zoom this down here, so we can see the sequence. So this whole bar represents the entire sequence. And if you zoom in enough, you'll be able to see the actual amino acid sequences here. And then you can drag this bar across to see which kind of annotations you would like. You can also have the filtered consequences and the provenance. If I want to look at this one, if I click on it, the table will scroll down to the corresponding section that will show the likely pathogenic annotation that I've clicked over here. If you go over to the feature viewer, this will also open a new tab. Um, Same sequence, you can zoom in and see the sequence itself. And once you're there, you can drag around and you can see uh, the tracks as well. So I'll open the PTM track, and here there's the modified residue in multiple areas. So I can click this uh, for each of the annotations, and you can see the description and the evidence from PubMed or your PMC, and you can explore these as you want. And once again, if I click this annotation, there's a table on the bottom that will take you to um, the area. So that is all good. I'll go back to the slideshow. And yes, so a quick recap of the changes in UniProt KB. So inside the entry tab, we have the cross-species AGR orthology data that is new. And within the structure section, we have the new AlphaFold isoforms and the complexes. In the variants viewer, we have the Canvas implementation, which is a bit faster than the previous implementations that we had. Annotations include updated PTM's annotations in the feature viewer. For the cross references, we have the card mechanism showing in the respective protein sequences that do have that contained. And also for history, we have the enriched history part that we've shown with the demerged and um, entries and the entire history as well. So that is it for UniProt KB. And then for UniParc, I will hand it over to Swati.
Hello, we'll now switch to UniParc. As Minjoon explained already, UniParc is a non-redundant database of protein sequences. Each protein sequence is taken from a public sequence database and it is assigned a UPI. It is a stable identifier. Uh, it is never changed or reassigned to a different sequence, so all of them are unique, and once it's there, they are preserved forever. In a UniParc entry, along with the sequence information, you'll also see the cross-reference table showing from where this sequence was taken from, when it was first seen, and when it was last seen, if it was ever deleted. So that is all in the cross-reference table. And the one thing is, in a month's time, we are going to remove all the non-reference proteomes from UniProtKB, and they will be moved to UniParc. So this is where you'll find all the non-reference proteome entries. Today, we'll touch the UniParc entry page and the newly added things in UniParc. If the one thing, if you search against UniParc, you can either choose to search against all sequences, or if you are interested in one particular taxonomy, you can use the advanced search tool and then you can search by taxonomy. In case of that, if you're searching by taxon ID, there is a suggestion that do you want to narrow it down to to that organism if the taxon ID is matching to that of organism ID. So you can narrow down your search to a small result set, and if you go to organism ID, you can even restrict it to proteome level. It will list down all the proteomes that are ever observed, like the video we have got reference proteome and a non-reference proteome. So it's just to help you navigate better and find the entries of interest for you. And this is also done in proteome namespace as well, along with UniParc, the same kind of taxonomy, organism, search suggestion, and vice versa. The next one we are going to see is if you search in UniParc for a particular proteome, our FASTA download is improved. The FASTA header now has protein name, gene name, like organism information, and cross-reference information in the FASTA header. As you see in the video, it is on by default, and we also recommend to use the same header because you have so much information there. Otherwise, you'll just have the UPID and the status. If you are accessing programmatically, in the same download panel, there is a link, there is a button called Generate URL for API. So you get what API query to run to get the enriched FASTA. So both website users and programmatic users can benefit from that. Okay, the next one. So this is a UniParc entry page. So as you see, there is an overview, and then there is entry tab, which has a sequence and cross-reference table, and then the new tab, which is structure viewer. Here we're using the sequence checksum and pulling out all the AlphaFold predictions that are generated for this particular sequence. So you'll see a big list there. This is to help to find the structures of sequence that you can't find in UniProtKB anymore. Then the next one is I'll show you the cross-reference table behavior now that we're moving more stuff here. We have made some improvements on UniParc entry page itself. The first one that are highlighted in green, they are actually active SwissProt and active Tremble entries. So if you click on those IDs, they will take you to the UniProtKB entry page. Because they are alive, and all the annotations that are rich, you can find them in UniProtKB namespace. If an entry is inactive and it is SwissProt, it will take you to the entry history page that Minjoon showed earlier. There you'll find more information. If it is deleted for so reason, you'll find the deletion reason. If it is more like if an entry is demerged into many entries, or it is merged into a new entry, all those information you can find in the entry history page. If an Tremble entry is inactive, Clicking on that link will take you to a new page, which I'm going to show you later. We call that sequence annotation page. That is a page dedicated to a cross-reference of UniParc entry, which I will explain later in the next slides. And then we have more links about external databases too, like RefSeq, Ensemble, etc. For them, if that RefSeq or Ensemble entry is active, it will take you to the new sequence annotation page, or if it is inactive, it takes you to the respective source database page itself, like it was before. This is the new sequence notation page that I told you about. The layout is quite similar to the UniProtKB entry page. So this page is whatever entry is being deleted in UniProtKB, you can find it here. Not only that, even we show the same page for the external source database too. You can see that there is a there is an overview and followed by a context that so and so that this entry is not is no longer in UniProtKB because it is not part of reference proteome. And if you still want to go to visit to see the history of that particular entry, you can still go to UniProtKB namespace, but only history tab will be visible because that entry is deleted in UniProtKB. And just like in UniProtKB, you can see the side navigation bar, which will take you to different sections. And then you can see that there is a tick with predictions generated. We are actually using our automatic annotation rules to generate predictions on the fly, and then we are sprinkling it all over the website. Um, so they are called unifier annotations. If we go to next slide, we'll see that they are basically uni rule and r bar rules combined together, and it is executed on demand. Like whenever you go and you land on a sequence annotation page, the annotations are run automatically, and you'll see the prediction generated. You can see that in the slide there is automatic annotation evidence tag, and on click it will be expanded, and you can see that is from actually a uni rule. That's how the function annotation is generated. This is helpful when the entry of interest of yours is deleted and you still want some information, and you can still see the annotations that will be generated by your automatic annotation, and you can still access them in the new page. Now we'll go through a demo. Let's search UniParc against all of sequences first, maybe. If you do a star search, so in this case, you're searching against all the sequences that are ever present in the archive. If I just want to look at taxon 9606, which is human, you see that, do you want to restrict the search to organism with taxon ID? Because there is an organism with that matching taxon ID. So it's asking you, do you want to restrict so that the other lower taxonomy ranks can be excluded? So if we click on that, you'll see either you can go up like where we actually came from, or you can restrict further to one of the many proteomes. In this case, we have got two. The first one is reference proteome, so it's quite helpful because those proteins are actually in UniProtKB. So they have more annotations curated by our curators. That's helpful to navigate from your UniParc to UniProtKB at proteome level. And the same will be observed in proteome namespace, as I explained before. If we search uh, by just proteome now, if we apply proteome filter in UniParc, if we go back and from the history, we can just grab the proteome ID. Yeah. I'll show you the FASTA download For this one, this should be noted that this improved faster header will be only available if you use only Proteome in your query. If you use this in combination with other query fields, it will not work for you. This is supported only for that one. And otherwise, you'll just get this Proteome UniParc ID with status without the enriched faster header. And in generate URL for API, you'll have for this particular faster header, which query to use, which URL to use. Okay, if we just go to the demo slide, we can grab the example for deleted entry. Yeah, that entry. If we search it in general search, like from UniProt itself, from the home page, I know that this entry is actually, yeah, if you search it, this entry is actually deleted. So if you have an entry that was deleted from UniProtKB, you will be redirected to UniParc where this cross-reference is observed. In this case, this entry is originally from this UniParc entry, and here you have all the unifier annotations generated and yeah, unpopulated. We have got function section, and then we have got more automatic annotations generated in names and taxonomy, subcellular interaction, and here as well, just like in UniParc entry, we have got AlphaFold 3D model prediction for even for deleted entries. This is again based on the sequence checksum. Okay. Yeah, now if we go to a different example from the demo slide, we'll just go through quickly about the cross-reference table. I pointed out that clicking on any active UniProtKB reviewed or unreviewed will take you to the UniProtKB entry page. And if we are clicking on a reviewed but deleted, it will take you to the entry history page. In this case, this is demerged into two different entries. And if we click on a deleted tremble one, this is again active. The B9, yes. Yeah, this is from Tremble, but it is no longer in ProtKB because of it was deleted from sequence source emble. And here again, you have got more unifier annotations generated. So this is how you can navigate to sequence annotation page. Either you come from the whole generic search. If the entry is not there, you'll end up in the UniParc sequence annotation page, or from the cross-reference table itself. So you'll see the same thing for an active external database ID. For this refseq ID, again, it will take you to the new sequence annotation page. So this is how you can navigate to that new page. Okay, and then we have got the structure viewer just to show you in UniParc entry page. In this case, there is only one. Okay. Okay, a quick recap would be, we have seen how to do a general search in UniParc with taxonomy query, with Proteome query, and we saw how to download FASTA for the Proteome query download. And we saw the structure viewer in both UniParc entry and sequence annotation page, and how to navigate to those pages from the cross-reference table and also from generic search. And we also explained how the automatic annotations in those pages work. It combines the unifier rules, which is UniRule and ARBA rules. Now we are moving on to tools. We'll specifically focus on BLAST and ID mapping today. If you go to BLAST form and paste a sequence that you grab from somewhere, and if that sequence has exact matches in UniProt itself, including isoform matches, it shows this isoform matches, and then there are more reviewed matches, like sometimes even deleted entries match your sequence. So all of them are included in that new table. And there is a link to view the matching sequence in UniParc, so which will also take you to the UniParc page. So this is helpful when you blast, you try to find a similar protein for that, but in which case if you want a really exact match, you don't even have to submit a blast request. You can just grab the proteins that will be helpful from here itself. And then this is the same behavior you can observe in Align as well. You'll have the similar table, or if you're just searching UniProtKB with the sequence, if you just paste your sequence in the search bar in the home page, it will show you the same thing, that these are the matching proteins that we have found, or these any of your interest. So this is common among in three places. And then the next one is in the blast form itself. Let's say that you have got a sequence and then you have chosen UniProtKB, RefProt, and SwissProt as your target database, and you're restricting by taxonomy. If you see in this screenshot, there is a zebra taxon selected, but the combination of these filters, the target database, your sequence, and this taxonomy, there is no matching sequence to search against, to blast against. So in that case, we just showed that there is no protein sequence to match, because your result will be zero even if you run your blast. Because of that, we disable it and show a warning. But if there is any match, we'll just show an information that you're going to blast against this many sequences based on the combination of your filters that you have used. And then you can submit a blast for that. This is an additional improvement, just preventing an unnecessary request that will yield no results in the end. This is the page of ID mapping results. In this case, I have submitted one active ID and then 6 deleted IDs. For the deleted IDs, we show that this has been deleted, and if you click on that link, it will take you to the sequence annotation page that we talked about earlier. And if you're interested in the IDs that are deleted, if you want, we offer you an option that you can still map these obsolete IDs to UniParc instead of UniProtKB. And you'll be able to find more information there. Okay, if we go for a short demo. We'll just grab a sequence. I'm going to blast against this sequence now. As you see that we have got this exact sequence matches an isoform, and it also matches the canonical and then other entries that share the sequence. So all of them are presented in line within one table. And there is also a link to the matching sequence in UniParc, or you can still see all the active entries in UniProtKB with the first link. And if we try to, yeah, okay, the whole search will also show you the same table, just like in Align and Blast. Oh, we need to paste one more. That's okay. If we go to Blast again and try a taxonomy, let's say 9606, or yeah. So it says that there are this many protein sequences you're going to blast against. If the filters don't match, if the taxon you have selected is very rare and nothing is common with the sequence, it will show you a warning like this, that this will not give you any results, and then you can see that the run blast button is also disabled. And then we'll go to an ID mapping job. There is a pre-run ID mapping job that we had in the screenshot. In this case, all of them are matching. If there are deleted IDs in the results, the same behavior, you get, there is an option to map them to UniParc and then clicking on any deleted entry will take you to the sub page, sequence annotation page. Yep. Okay. These are the tools improvements. Yeah, we saw how to use BLAST and then the improvements made there, and then from ID mapping, if you're lost, how you can redirect it to UniParc and still you can find your entries records there. Throughout the website, we have got our help documents inline, and you can find the help at the top right corner where help and contact are together. And all the recorded training and webinars, you can find them in our YouTube channel. And if you want to look at the release notes for every release, what is changing, you can follow that link there. It will show you all the release notes of our previous releases and forthcoming changes that if there are any breaking changes in upcoming release. You can always contact us, leave feedback to us in that following mail ID, or you can also go through to our contact form and then you can type your message. We'll receive it.